while we are running, that's fine. We can continue. So, we did go through epidemiology last week, correct? And we did fast QC and multi QC. And the idea there is to get a sense of the quality of your data. Stanislav, so I will help with some translation at some point. Okay, no worry. So today, what we want to do is to go through quality, because quality is important. Okay? So the first thing we want to ask ourselves is, why is quality important? Secondly, we want to look at quality in terms of two things. Looking at read quality, which is what you read using fast QC, and two, using assembly, looking at assembly quality. Because when you get a consensus file, you also have to assess whether the consensus sequence you're getting is good quality or not. Right?
Donc, ce qu'on va regarder aujourd'hui, d'abord la première question qu'on se posera ici, c'est de savoir pourquoi la qualité est importante. Et donc on va regarder la qualité sous deux aspects, c'est-à-dire la qualité des reads et puis la qualité des assemblages. La qualité des reads, quand vous avez vos données qui sortent du séquenceur, vous devez regarder d'abord la qualité pour voir quel est le niveau de qualité de vos séquences en sortie du séquenceur. La deuxième chose qu'on va regarder, la qualité de l'assemblage réalisé. Quand on fait je construis un génome consensus, on a besoin savoir que ce génome consensus-là est de bonne qualité. Donc, c'est ce qu'on va essayer de regarder aussi aujourd'hui.
All right. So, how many have worked in the lab? Good, right? So, you are familiar with this process. So the first thing is you do your extraction, then you do library preparation, right? Then you do sequencing, right? And then after that, you do bioinformatic analysis, which is what we are training for, and then genomic epidemiology, right? So up to this point, we are assuming we've done all these steps. You're getting into the bioinformatic analysis, and then this morning we'll also start on the genomic epidemiology. Okay? Now, we also want to think about the steps in bioinformatic analysis. Okay. So, we've looked through these steps. Okay? Now we want to go here. Okay. So, in the bioinformatic analysis, we have what we call data pre-processing. And you'll remember that Peter went through this as one of the stages that happens within epidemiology. So one is you remove low quality reads, okay? You remove adapters, and also you trim sequences, okay? So that you get reads that are of within given quality, okay? Secondly, you do your assembly.
Ok. Donc, ce qu'il vous a montré précédemment, ce sont les étapes, c'est-à-dire les étapes wet lab et les étapes dry lab, c'est-à-dire les étapes de bioinformatique. Donc, quand on s'intéresse principalement aux étapes de bioinformatique, il y a plusieurs étapes. La première étape qu'il a présentée, la première étape, c'est l'étape de data processing. C'est une étape qui est très importante parce qu'elle va nous permettre de supprimer les reads de mauvaise qualité, elle va nous permettre de sortir les adaptateurs, et puis elle va nous permettre de trimer nos séquences, ce qui va nous permettre en fin de processus de data processing d'avoir des reads de bonne qualité. Et après cette étape-là, on va procéder à l'assemblage. Si vous voulez l'assemblage, il faut regarder l'assemblage comme la construction d'une maison. Lorsque vous voulez construire une maison, vous partez des briques que vous assemblez pour avoir des murs. Donc on a des reads, on les met ensemble pour avoir ce qu'on appelle des boutiques. Et après, on peut mettre ensemble des boutiques pour avoir des éléments plus grands par la suite des scaffolds. Donc cette étape, c'est l'étape d'assemblage.
Thank you. So, as is explained, you could assemble your reads into either contigs, if you're using de novo sequencing, or consensus, if you're using reference-based assembly. Okay? Then we do have a notation where you have your assembly, and then you want to like annotate your consensus with like which part of the sequence is a gene, which part of the sequence is not a gene. Then we have a step where you do genomic characterization. So this is where like you for example want to know the lineage, clade of the sequence and those kinds of things. So for example, this is what we did with NextClade. And then lastly, you do some phylogenetics, so you get a phylogenetic tree. Okay? So this is a typical process that we undertake in bioinformatic analysis.
Donc, après les étapes d'assemblage, vous serez souvent amené à faire ce qu'on appelle une annotation. Alors, une annotation, c'est de retrouver tout ce qui est variation génétique et prédire leurs effets, c'est-à-dire leur impact fonctionnel. Après cette étape, on peut s'intéresser à certains éléments génétiques et essayer de lier ces éléments génétiques avec certains aspects phénotypiques tels que la virulence et autres. Et après ces aspects-là, on essaie de regarder l'histoire évolutionnelle, c'est-à-dire les relations entre l'histoire évolutionnelle entre les différentes séquences.
Let's get a question. Anybody with a question? Any questions? Good? Okay.
I have a question.
Yes.
I want to say, when you use the processing of that, first you said that the objective to remove the local phenoids. But then at the end you said that so you can trim the sequence. Is it different or trim sequences? You remove the low quality sequences. Is it a full trimming or it's a
Now, they can be independent, but they can also be related. Okay? Let me explain why. Now, when you give your reads to a workflow, again, remember that workflows are different. Okay? And we're going to talk about quality. Now, we're going to talk about, let's say, do you know, I'm going to show you shortly what that is. Do you know Fred score? Like, which we say Q equal, let's say, 30. Okay? So let's say in our workflow, we are saying that we are not going to take any sample whose mean quality is less than 30. Okay? So what the workflow will do is that any sample with mean read quality of less than 30 will be thrown away. Okay?
Okay. Ce qu'il explique ici, c'est le trimming des échantillons. Le trimming des échantillons. Lorsque vous avez vos échantillons, l'une des questions que vous pouvez vous poser, c'est que vous pouvez dire par exemple, je vais prendre un threshold, c'est-à-dire un seuil. Chaque séquence a une qualité moyenne, exprimée en ce qu'on appelle en qualité de frais, ou des scores de frais. Et donc on peut se dire par exemple, dans mon échantillon, où il y a plusieurs rues, je peux décider de regarder que ceux qui ont une qualité au-dessus de 30. Donc, dans l'étape de préparation de nos données, on va peut utiliser un outil qui va supprimer tous ceux qui sont au-dessus de 30 et garder tous ceux qui sont au-dessus de cette valeur. Donc, ici, l'exemple qu'il a donné, on viendrait donc filtrer sur la qualité moyenne des séquences.
So here we've not done any trimming. So if we set our quality here to say, let's call once, oops, so greater than or equal to 30. So this sample will not go through, right? This one as well, right? And then we know we have to remove primers, okay? So you remember last week Peter talked about removing primers from sequences. So always you have your read, then you have like could have primers there. So you have to remove those bits, okay? Now what the workflow does is every time you remove something, then it recalculates the quality, okay? All right, so now we've removed our primers. We ask Q equal what? Then you get that quality. Maybe after primer trimming, this goes to, I don't know, could be, guess, it could go lower, but who knows? Let's play around with this. So we could say this goes to 35. Could even say this goes to 29. This goes to 40. Okay? So we trim primers, and this one, after removing the primers, the quality goes up a bit. Okay? This one, the quality goes down. This one goes up. Okay? So now, again, this is lower than 30. We remove that. Okay? Now there is the last step where, you know, you want to trim the read. So there are two things. Have like this.
I have access to it. It's in the folder.
Was it saved here?
No.
Okay. Yeah, but
It's deleted every time the file goes off.
okay. But thank you. You remember this? You remember this from last week? So we are just going through. So there was this stage where you say you trim adapters, and now we want to go to. So let me just write on this side. So first, remove low quality reads. And we are doing this even before we do anything, right? The reason we are doing this fast is because we do not want to waste time with reads that are just of bad quality. There's no reason why you should be removing primers from a read that is already bad quality. Okay? There's nothing that's going to change.
Ce que vous expliquer, ça c'est une étape très importante dans votre formation. Quand vous disposez de vos séquences, il y a une question principale. La première question que vous allez poser, est-ce que les séquences sont de bonne qualité? La deuxième chose que vous voudrez, c'est de continuer vos analyses avec des séquences de bonne qualité. Donc, c'est la toute première chose que vous serez amené à faire. Donc, vous allez filtrer sur la qualité s'il y a des primers qui sont classiques. Ces primers, en fait, représentent du matériel génétique non d'intérêt, n'est-ce pas? Donc, vous allez les enlever aussi.
The second step, we remove primers. Again, we check if the Q value is below 30 or above. Okay? Then we continue. Then the third step is what we call clean reads. So, I'll skip this a bit. So let me come back. Is it HDMI one or a fish? Oh, wait. Okay. Okay. Now, if you look at your first QC outputs, right, you'll notice that towards the right, at 3, you can see that this line here goes down, right? This is the mean quality, okay? You'll see that this goes down. And this is normal for sequencing even back then when we still used to use Sanger sequencing. It's just based on the physics of how the bases are called. Always you'll have low quality, especially at the 3' end. Okay?
Ok. Donc, si vous regardez, quand on a fait le phase QC, l'une des sorties, c'est celle-là. Et sur cette sortie-là, ce que vous voyez ici, c'est la qualité moyenne des rues. Une chose que vous devez retenir, c'est que généralement, enfin, sur la section 3', on a généralement des bases de mauvaise qualité. Donc, ce qu'il faut regarder ici, ce que vous voyez ici, ce sont les scores de fait. Dans le rouge, c'est des trucs, c'est des séquences de qualité moyenne, de mauvaise. Ici, c'est acceptable, et puis ici, c'est de bonne qualité. Donc, vous voyez, on va chuter comme ça. En moyenne, je veux dire. Et donc, parce que c'est un outil qui va nous permettre d'avoir un regard visuel de la qualité moyenne de nos séquences.
Okay? So let's say you have a sample that has a Q value after trimming primers. Maybe the Q value is 28. Okay? So at this stage, what we want to do is, and at the end here, we have, let's say, the quality at the end of that read could be like 24, right? So what you do is you try and remove that base, and then you calculate the mean quality and see if it goes above 30. If it's not, you remove the next base. And you continue like that until you get a mean quality of 30 or above 30. Okay?
Supposons par exemple que nous avons à la fin, dans la section 3' ici, des zones avec des qualités moyennes en dessous de 30, disons peut-être 24. Nous, ce qu'on recherche, c'est quoi? On a fixé un threshold de 30, n'est-ce pas? Donc, si on fixe un threshold de 30, qu'il y a des bases qui sont en dessous de 30 vers la section 3', qu'est-ce qu'on va faire? On va couper, n'est-ce pas? Donc, on va dire qu'on va trimer dans la section 3' et on va recalculer. Et chaque fois qu'on aura des bases en dessous de notre threshold, on va continuer à trimer. Qu'à la fin, on n'a que des bases qui sont quoi? On a des reads et des bases, quel que soit l'endroit où en qualité moyenne, quand on refait le calcul, il faut qu'on soit autour de 30. 30 ou au-dessus, je veux dire.
Okay? But, let's say you've done Illumina sequencing. What is the typical length for Illumina reads? 150? 151? Or sometimes you can do the 300 cycles, so it could be 300, right? Okay, let's work with 151, okay? Let's say you continue trimming, but you also have to think about your minimum read length. Because what if you trim and you only get above 30 when you have 10 bases? Is that useful? No.
Supposons que quand vous prenez les individus, la longueur standard c'est 150 mètres, n'est-ce pas? On se retrouve dans les on peut calculer pour avoir 300 mètres. Supposons qu'on a 100 mètres. Je coupe. Au fur et à mesure que vous coupez, c'est vrai, la qualité monte, mais qu'est-ce qui diminue? La longueur de la séquence. Supposons qu'à la fin, vous vous retrouvez avec une longueur de 10 paires de bases. Voilà. Donc, il faut trimer, mais il faut tenir compte aussi de la longueur dont vous trimez.
Yeah, so you also have to decide what is my minimum length.
Vous devez décider aussi qu'il y a une qualité moyenne des runes, mais il y a une longueur moyenne aussi qu'il faut tenir. Donc, ça fait déjà combien d'éléments? On en a pour 2.
So, for example, a lot of people would say, okay, maybe 100 or 70, people decide different things. It's something we'll talk about, like which standards should you use. So let's say you say my minimum read length is 100, okay? So if you are chopping the bases at the end and you reach 100 and the quality is not above 30, you stop and you throw that read out.
Si vous décidez par exemple que votre longueur minimale moyenne, c'est 100, au fur et à mesure que vous prenez jusqu'à atteindre ce threshold là, et la qualité n'est pas remontée au-dessus de 30, qu'est-ce que vous faites? Tout le rythme vous le dit.
Okay? Yes. So these are very important metrics that you have to think about when we are doing this. So I'll go back to my slides. So, data expectation. What do you expect your data to look like? Very beautiful, yes? Right?
Ce que vous espérez, c'est d'avoir des séquences de très bonne qualité.
But always, sometimes your data looks like that.
Malheureusement, dans la réalité, vous allez commencer toujours avec des séquences de mauvaise
Right?
qualité.
So, I like this. Garbage in, oops, garbage out. How do you say that in French? Do you know what garbage? Garbage, c'est ça.
Donc, généralement,
What is it?
en bioinformatique, on a l'habitude d'utiliser ce terme-là.
Qu'est-ce que c'est ça en français?
Si vous mettez de la sanité dans votre pipeline, vous allez ressortir avec des résultats qui ressembleront à la santé.
Si vous utilisez data
de
comme
mauvaise qualité,
très mal,
vous aurez des résultats de mauvaise
the results will also be bad. Okay, that's what that means. Bad data, bad results. Okay? And you see, if you use bad data, you can't also get strong conclusions from your data because the data is bad, right? So that's why we say garbage in, garbage out. And that's why you see the cleaning and pre-processing of data is very important. Okay? So that you're trying to use only good quality data. Okay? Because if you use bad quality data, you might say, oh, I have a new mutations. I have very nice mutations. But then later, you go and look at that data, and it's pretty bad. And you don't have new mutations. Because the quality is so bad. Right? Yes. So we always want to use high quality data. Okay? All right. Now, we have talked about a few quality thresholds. For example, Q30, minimum length 70 or 100. The question is, who sets this quality thresholds? Who decides?
Okay, donc on a parlé de plusieurs thresholds, n'est-ce pas? La question, c'est qui a fixé ces seuils?
So the truth is, a lot of the quality thresholds have not been set for many pathogens. Okay? For some pathogens, you do have some quality thresholds. So I don't know who works with bacteria. Who knows PulseNet? I think he knows PulseNet. So PulseNet has quality thresholds for some pathogens. Some, not all. Okay. So what often happens is that you would need to define the quality thresholds in your lab. But these quality thresholds have to be defined against reasonable biological and technical expectations. Secondly, they have to be specific to the organism and the sequencing approach or technology. Okay?
Donc, généralement, lorsque vous voulez fixer vos thresholds, il faut tenir compte d'un certain nombre d'éléments. Vous devez tenir compte des aspects biologiques de l'organisme que vous utilisez. Vous devez tenir compte des conditions techniques. Et aussi, il faut aussi regarder l'organisme et la technologie de séquençage que vous utilisez. Donc, généralement, quand vous voulez fixer vos thresholds, ce que je veux dire, les thresholds ne sont pas totalement définis pour tous les organismes. Pour certains organismes, c'est défini. Mais si, quand vous voulez fixer vos thresholds, dans votre lab, vous devez tenir compte des aspects comme les conditions biologiques, les conditions techniques, l'organisme que vous utilisez, et puis la technologie de séquençage.
Okay? I will give you an example. How many have done ONT sequencing? Okay. So what is the usual quality for ONT reads? 10 to 15. Eh? 10 to 15. So do you think if I decided to say my minimum quality is 30, do you think that will work? Because it is not reasonable for the sequencing technology, right? So as you think about these quality thresholds, you need to think about the organism, the sequencing technology, and also just the biology, okay? Now, the challenge here is there are different organisms, okay? You're not going to treat, for example, RNA viruses and DNA viruses the same way. Because some are more difficult to sequence because maybe they have repeat regions, maybe, some are longer, some are shorter. They're different, okay? And, you're dealing with, there's also bacteria, there's viruses, all those kinds of things. Secondly, the sequencing approach. You've done whole genome sequencing, you've done metagenomics, for example, right? They're different. The quality thresholds are going to be different, okay? The technology, I've given an example of maybe like ONT. You have PacBio, you have Illumina, you see, different technologies. And also the use case, right? Is this for research? Is this for diagnostics? Is it for, a clinical case? What are you doing? See? So all these things will determine which QC criteria you use. And you can see how it's difficult. It's not easy. Right? Okay. So again, you can decide which quality to use, but remember, it has to be informed by the organism you're working with, the technology you're using, and what you want to use the data for. Okay? All right, so this is the structure of a fast Q file. All right, first Q file usually have the name, we have the sequence, we usually have that plus sign, and then we have the quality.
C'est vous comprenez les fichiers. Ok. Lorsque vous faites le séquençage, généralement, que ce soit Illumina ou Oxford dans le bord, la principale sortie du séquençage, ce sont les fichiers FASTQ. Compression ou non compression. Vous avez vu, on a des FASTQ ou FASTQ GZ. Mais qu'est-ce qu'il y a à l'intérieur? On a des RUID. Mais les RUID, d'un point de vue organisation, se présentent comme ceci. Donc, sur la première ligne, généralement, un RUID est représenté par 4 lignes d'information. Sur la première, il y a l'identificateur du RUID. Sur la deuxième, il s'agit du RUID lui-même, c'est-à-dire la séquence. Après, il y a un signe plus. Et vous regardez ici, ces caractères qui sont là, c'est ce qu'on appelle les scores de qualité, codés en caractères ASCII. Et ce sont ces caractères-là qui vont nous permettre d'avoir notre score de frais. Donc ici, par exemple, si je prends le S, on va dire que sa qualité, c'est H. Et le H, ça correspond à un score de frais bien défini. Donc,
Each character has a value. So maybe H, maybe, for example, means 10. Okay? And you see, petit h and grand h, they mean different things. Okay? And even this one has a quality encoding for it. Okay? So this is how a FASTQ file. I mean, the tools that we use, they understand. They will know how to convert this into the Q value. Okay?
Les outils qu'on va utiliser pour ça, pour convertir ces caractères spéciaux, les caractères en Q value. La qualité des bases, les outils savent comment les traduire en valeur numérique et puis utiliser ces valeurs numériques en calories pour avoir une qualité value. Il y a des formules aussi qui permettent de les recalculer.
Okay. You can see here, for example, we have, here is a character, okay? This character
Point d'exclamation.
has been assigned a quality value of 0.
Donc, point d'exclamation, si vous regardez la colonne de Q, point d'exclamation correspond à 0. Donc, si vous avez une base qui est marquée, par exemple, où la qualité c'est le point d'exclamation, ça veut dire que sa Q value, ce sera 0.
And you can see that, you know, different characters here have different quality scores. Okay? So that's why when we go back to our slide set here, if you look at this read here, they will have, you know, each shark base has a value. Okay? And then you go to the next read and like that. And as usual, you expect one file to have thousands of reads. All right? Okay. No, this one doesn't mean anything. I think it was just used to distinguish the two lines. All right. I also just wanted to show a FASTA file which only has a header and the sequence, no quality information.
Donc ici, le fichier qu'il vous montre, c'est un fichier au format FASTA. Si vous regardez le fichier précédent qui est au format FASTA, la ligne d'identification commence par arrobase. Pour le FASTA, ça commence par le signe supérieur. Et dans le fichier FASTA, on a l'identificateur et la séquence. Il faut bien connaître l'organisation de votre fichier. Pour le FASTA, voici l'organisation, c'est l'identificateur et la séquence, alors que pour le FASTA, il y a 4 éléments qui identifient un nom.
Okay. All right, so we've talked about a lot of these things. We've talked about the Fred quality score, and if you have a read quality score of 10, it means that, so it's a probability calculation. Yeah? So it's a probability that 1 in 10 bases is called incorrectly. Okay? And a Fred score of 20 means 1 in 100. Now, there's a misconception that people have and think that after every 100, for example, for 20, that base is wrong. That's not correct. Okay? And as we've seen, a read file, often the read quality is bad at the end. So when somebody says that, oh, you have a Fred score of 20, which means 1 in 100, it does not mean you start from position 1, and then number 100 is bad, and then you go number 200 is bad. No. This is a calculation of probability over the whole read. Understood?
Ici, c'est qu'on essaie de comprendre ce que les valeurs de quality score veut dire. Si j'ai un score de qualité de 10, ce que ça veut dire, c'est quoi? C'est que généralement, lorsque vous faites du séquençage, il y a un outil qu'on appelle le base scorer. C'est lui qui fait les appels de route. Et donc, lorsqu'on a une qualité de score de 10, ça veut dire que la probabilité d'un mauvais calling est de 1 sur 10. Donc, ça veut dire que la précision ici, c'est pratiquement 90%. Donc, si on a un score de qualité de 20, ça veut dire que la probabilité d'un mauvais calling, c'est de 1 sur 100. Ainsi de suite. Il faut bien retenir que c'est une probabilité qui est calculée sur l'ensemble des routes.
So we always like 30 because, you know, it's 1 in 1,000 and you have an accuracy of 99.9%. So that is good. So most people use 30. Okay? But again, we said, you know, you could decide like I'm going to use 25 or I'm going to use 35. Okay? But it has to be, you know, you have to know why you're making that decision.
Généralement, on utilise 30 pour ce qui est là. Ça veut dire qu'il y a une probabilité d'une base incorrecte sur 1000, ce qui nous donne une précision de 99,9%. Mais vous pouvez décider de prendre des sauts. Ça dépend des conditions, de vos conditions propres. Il faut prendre vos décisions de manière. Il faut que vous ayez des éléments qui vous conduisent à décider de choisir certains choix. En vous basant sur ces éléments pour
So we've talked about this. We've said usually towards the end, the read quality is lower. And also secondly, because, you know, we typically do paired end reads, right? Forward and reverse. The reverse read is generally usually of lower quality than the forward. Again, just goes back to the physics of how the sequencing works, right? So expect that your reverse reads will be of lower quality than the forward. Okay? Now, we talked about looking at the Q value, looking at the minimum length. Sometimes, some groups also decide like, okay, after I've done all my read trimming and everything is quality above 30, they also look at the ratio of the good quality reads in the forward and the reverse. Because, for example, you might have that your good quality reads in the forward, they are 80%, and the reverse, they are 20%. Or you could have like 60-40, Which one is better? Like your forward reads, good reads. Forward 80, cut 4. Reverse 4. And in another sample, forward was what? 60, and the reverse 100. Which one is better?
Ce qu'il essaie d'expliquer, c'est que généralement, lorsqu'on fait un séquençage de type immunal, on peut décider de faire une single ou bien c'est-à-dire séquencer dans les deux sens. Mais il faut vous rappeler, c'est que si on fait un séquençage pérenne, ça veut dire quoi? Ça veut dire que si sur, par exemple, le grain direct, les séquences sont de mauvaise qualité dans le sens droit, sur l'autre grain, ça va mauvais dans l'autre sens. La question qu'il pose, si j'ai par exemple 20 et 80, ou bien 40 et 60, qu'est-ce que j'ai choisi comme séquence de bonne qualité?
Aussi, la séquence qui a le sport dans les deux sens, en fait.
Dans les deux sens?
On n'est pas toujours obligé de travailler avec le grand sens et le petit sens.
Non, mais si tu as fait un séquençage pérenne, pourquoi tu vas décider de choisir de prendre un seul grain? Quand on décide de faire, oui. J'ai 80-20. Et puis de l'autre côté, j'ai 60-40. 80-20. Et puis de l'autre côté, sur l'autre route, j'ai 60-40.
Pourquoi?
Pourquoi tu prends 60-40?
Parce que dans 80, voilà, c'est comment on appelle ça? Ça va. Si c'est mon. Comment on appelle ça? Ma longueur, c'est disponible, ça va prendre le chef.
No. Remember that both forward and reverse cover the whole genome. So, deciding between 80-20 and 60-40, you have to ask yourself, why do we do paired end sequences? Why? It's because we always want our assembly to be as accurate as possible. You don't want a scenario whereby if the forward reads are making an error, right? You want to be sure that, if the forward reads are calling a position A, the reverse reads are also calling that position A, right? Because you might end up with a situation whereby the forward reads are calling that position A, but the reverse are calling it G. That means there's a problem, right? So always you want a similar proportion. So 60-40, that is good. 80, even if we know the reverse, generally the quality is low, but we still want a similar proportion of the forward and the reverse. Understood? Yes. Okay. Perfect. Yes.
Yes, I get on that, but then because you understand the other thing that they are doing is because I believe probably that every correction step we are not as accurate as possible. So we are looking at when you have produced a family site that is more then the forward read will not cover every time. So that what then comes in with other side.
Well, at least for most tiled amplicons, you expect lots of overlap, correct? So you're right. I mean, generally, this is what we expect to be where you have tiled amplicon sequencing, and some degree of overlap is expected. If you're right, I mean, if the insert size is pretty long and there's very little overlap, then that's I think that goes back to what you were saying, that you have to have reasonable expectations based on the technical conditions in which you're sequencing. So this is just showing what we've talked about. So at the start, you have your reads like that, forward, reverse, and then we remove low quality reads, we remove low quality ends of reads, and we also remove adapters. So essentially, you end up with something like that. Yeah. So for some of the pairs, they will be retained. You can see these ones have not even been trimmed. Maybe there were no low quality sequences, there were no adapters in those reads. So they are good, right? For this one, the reverse was bad quality and it was thrown out. For this pair, both of them were bad, went out. Okay? So this is what you expect after the read cleaning steps. Okay?
Ok. Donc, lorsque vous allez faire du trimming, généralement, vous allez supprimer les bases de mauvaise qualité, les bases avec de mauvaises qualités sur les extrémités, et surtout, vous allez enlever les adaptateurs. Donc, après avoir effectué l'étape du trimming, vous allez voir que vous allez perdre, que ce soit dans les sens forward ou reverse, vous allez perdre. Et ce qui va rester, ce sont les mauvaises, de bonne qualité.
Okay. Now, always you want to connect the bioinformatics with the lab, right? Because it's important. We want to know if we end up with bad quality because of something that you're doing in the lab, right? So for example, your number of reads, very small. Okay, what does that mean?
Supposons que vous avez, en dehors, après ce séquençage, le nombre de reads qui est faible par rapport à ce que vous vous attendez. Qu'est-ce que ça signifie?
So maybe if you have too few reads per sample, it could be because you're multiplexing too much. You want to sequence too many samples at the same time, right? And maybe if you have too many reads per sample, it's too much. Then maybe it's because you're sequencing too few per run. But you see, if you have too many, one, it's going to be a waste, like computational. You're going to spend so much time, or you need to downsample. You would rather use that time to sequence a bit more per run rather than downsampling, right? But then again, it depends on the objective of what you want to do. If you want to call variants, and that is very, very important, then you always want as high coverage as possible, right? But then you can also be sequencing still too much. You just need to estimate okay, do I need a coverage, for example, for 200? And you see, this is what typically happens in the lab. Even in the lab, you always have to optimize your sequencing. You do a run, maybe you say, okay, I'm going to do 48. You see the number of reads and the quality that you get, right? Maybe you do another sequencing, you do 64. You see how the depth and all these things happen. And you get to a point where you're comfortable, okay, if I sequence this number of samples, then this is the coverage that I expect, and this is the quality that I expect. Okay? Now, the second thing has to do with average read quality. If it is too low, maybe your library preparation is bad. Okay? Maybe, you don't remove all the, what do we call this reagents that, during, because always you do cleanup in library preparation, right? Maybe you don't remove enough of the ethanol, for example. And we also have average read length. So if it is too low, maybe your library is breaking too much. You expect, maybe 300 or 500, but maybe you're using too much energy, or the technology you're using is breaking your, your amplicons too much. Okay. Again, we should use, these quality outputs to help us, improve the laboratory process. Okay? Here you can see. Yes.
Qu'est-ce qui se passe par exemple lorsque nous avons bien préparé nos librairies, nous avons bien respecté la procédure, mais maintenant au niveau de la normalisation, finalement la planification qui est la fonction de normaliser sais pas. C'est-à-dire que, imaginons que lors du poulet, on ne fait pas une division assez correcte. Donc, lorsque peut-être la division assez concentrée, qu'est-ce qui va se passer à ce niveau? Qu'est-ce qu'on peut voir à ce niveau? Ou alors au cas où la division est assez très prononcée, lorsqu'on a plus une copie, maintenant on a dilué, puis c'est vraiment très dilué. Qu'est-ce qui va se passer?
During normalization. You see, we do normalization because we have a target, like the reads that you're given. The reason Peter did the normalization, one, was we are expecting that the computers you have small RAM. And these were very big files. And if you know how computers work, is that it takes a sample, and while it's working on it, think of it like it has to hold it in memory. So when it is holding it in memory, it's using RAM for holding the sample. Now, there is the RAM that is also needed to actually process the file. Okay? So if you have a big file, let's even say 2 GB. Your computer has 4 GB. Already 2 GB is gone. Now, it needs to process, for example, remove adapters, how much RAM is required for that. And remember, at the same time, your computer still needs RAM, to have your screen on, to have, your antivirus running in the background, to be doing a couple of things, okay? So that's why we needed to normalize the reads to a lower sample, but still, it's still representing the whole genome and also like a varied quality. So often, if you use a good tool, you don't expect any problems. Okay? Even though the normalization process is a bit random, but it's usually the solution to having, like, if you have too much, and you just need to reduce it. Let's say if you dilute your sample too much, it means you have little starting material and you're going to expect that there will be a low number of reads because you don't have enough, right? So if you don't have enough, it means that even as you're doing your amplification, that PCR process, you're just going to end up with very few. Yeah, and it's the opposite of if the quality of the quantity of what's going in is too much, then you're going to expect too much as well. But remember, there is also a limit just based on what the kit can produce, right?
So I think sometimes it comes to an issue when we are trying to pull together different samples. Then you have to decide, should I pull a COVID abnormality or is it product? And samples are really, let's say, given maybe the curve when they are totally different, so you have to decide on how to. And then some you find on your according to your coding method, some samples will become the strategic stuff like that.
True. I mean, the best case scenario is you expect all samples to be similar concentration so that you don't end up with that bias where some samples are sequenced more than others, right? But sometimes, even if you are the same concentration, there's just something beyond you, especially if you're working with different pathogens. Some pathogens are, automatically usually more, they'll just be sequenced more or faster than other samples. So, again, I think this is the reason why we go back to looking at this quality so that, it can inform our lab process. I think before we go for tea, I wanted to look at assembly quality, right? So we've talked about read quality. So let's say we have good quality reads. We've met our consensus or contigs, depending on the method that you use. Remember, we have two ways of doing assembly: de novo and reference-based. We can talk about that later. Okay? Yes.
Suppose, il faut bien comprendre ce qui va engager. Généralement, lorsqu'on regarde la question de la qualité, on regarde la question de la qualité en sortie de séquençage. Et puis, nos fichiers fassent que c'est ça. Donc, on vient de traiter cet aspect-là. Maintenant, on a des séquences de bonne qualité. Selon la technique, selon qu'on dispose d'un génome de référence ou pas, si on dispose d'un génome de référence, on va construire un consensus. Si on ne dispose pas de génome de référence, on va faire des assemblages pour arriver au côté de scaffold. À ce niveau aussi, quand on a notre assemblage, il y a une question qu'on va se poser: quelle est la qualité de cet assemblage? Et donc, la question qui va traiter maintenant, c'est la question de la qualité de l'assemblage.
Okay? So, one of the things, let's say you did de novo assembly. So, always with de novo assembly, you get contigs, which are like short fragments. Okay? Now, if you think about the number of contigs, you want few. It means that your assembly is not broken, right? Because if you have, let's say, how long is this? 100 plus, this is 130, right? 230, 330.
Yes. Supposons par exemple, quand vous voulez faire de l'assemblage, vous l'assemblage de niveau, ça veut dire quoi? Vous allez prendre vos routes que vous allez transformer en config. Donc ça veut dire que vous allez vouloir que vos routes soient là pour que vous continuez. Vous avez un nombre réduit de contigs, n'est-ce pas? Supposons que vous avez. Ce qu'on attend, c'est d'avoir très peu de contigs. Supposons que vous avez un grand nombre de contigs. Qu'est-ce que cela signifie? Donc c'est ce qui va être.
Okay. So just remember, de novo assembly, you will not get one assembly. Okay? Consensus, you'll always get one. And the only difference is that for consensus, what it does is, for the positions of the genome that you could not sequence, it adds ends. You remember the ends that Peter talked about last week, right? Okay, so let's assume that our genome is 400 bases, like here. In this case, we have 7 contigs, right? But maybe for the same sample, let's say if we had 15 contigs, which one is better?
Supposons par exemple qu'on sait qu'on veut construire un assemblage de mouvements, je veux dire. Et puis on a un où on a 7 contigs, et on a un autre où on a 10 contigs, ou bien 20 contigs. Quel est le meilleur assemblage?
Yes, you want it lower, because it means the sequencing is good, the coverage across the genome is good, so you have few breakages. Okay? So fewer is better. So for this, there's a tool that we call Quast that probably we can run after T. We see the kind of output. So it will normally give you the number of contigs. I mean, we've done consensus assembly, so the output here is always going to be 1. Okay? So for consensus, we can look at a different metric. Okay?
Il y a un outil qu'on appelle Quast qui nous permet de regarder le nombre de contigs. Mais nous, dans le cas d'espèce, ce qu'on a fait, on a généré des génomes consensus au niveau des contigs. Donc, ici, Quast ne va pas nous. Dans le cas des génomes consensus, on va avoir un seul. Et si on avait utilisé l'approche basée sur la construction des contigs, on va utiliser Quast pour regarder le nombre de contigs qu'on a.
So because for consensus, what we are going to do is to look at the proportion of the number of ends, which tells you how many bases would not be called from the reads.
Pour les génomes consensus, ce qu'on va regarder pour la qualité, c'est le nombre de N sur tout notre génome. Donc, si on a beaucoup de N, ça veut dire quoi? Ça veut dire qu'il y a un problème.
Oops. Oh my Okay. So the other thing is assembly length. So of course you want your assembly length to be as close as possible to what you expect. So if your assembly length is 400, you want a number that is as close as possible to 400, maybe 350, 380, not, you know, 200 or less. Okay?
Moi, généralement, je cherchais des assemblages qui sont propres de la nouvelle dimension.
Now there's something that we call N50 that is usually a bit difficult to explain, but I'll try. So this is the length of the shortest contig needed to make 50% of the length of the genome. So, okay, we know that our genome here is 400, right? So half, 50% is 200, right? So, and we start from this side, we start with the longest contigs. So how many of the longest contigs do we need to make 50% of the length of the genome? So here we need contig 1, which is 100, plus contig 2, which is 70, so that's 170, and half of this, which is contig 3, right? So we'll only pick like 30 here to make 200. So the length of the shortest contig that is needed to make 50% of the genome length, in this case, is 60. So you always want this number to be as high as possible. Okay.
Il y a une métrique qu'on utilise pour établir la qualité des assemblages.
Because when it is small, it tells you that your contigs are quite short. But when it's big, it tells you that, well, you have good contigs that are quite long. Okay?
Et cette value-là, c'est ce qu'on appelle le N50. Donc le N50 va vous dire si vous avez des contigs qui couvrent une bonne partie de votre génome, c'est-à-dire avec au moins 50% de votre génome. Donc la métrique N50, ce qu'on va aller chercher avec cette métrique-là, c'est qu'elle soit beaucoup plus élevée.
All right. So one last thing is the GC content. You know, in DNA we have A, T, G, C, right? So again, for your GC content, you expect this to be as close as possible to your taxon or the pathogen that you're working with, right? We do know that some organisms are AT rich, which means they have more AT than GC. Okay? Some organisms have fewer AT, right? Again, you remember, you know, there are purines and pyrimidids, right? That's why we go look by AT and GC. Okay? So, for example, in this sequence here, the number of G's and C's are 9, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. And the total length, 20. So our GC content or percentage is 45%. Which means if I know, for example, that, you know, this is an example, Vibrio cholera is 47.5, right? And then let's say this is Vibrio cholera and I'm getting 45. Then I have to be worried, right? Because why is it, you know, 2% is a bit worrisome for the sequence, right? But then again, you also need to think, sometimes our genome is not uniform, right? There could be sections that are AT rich, and there are sections that are GC rich, right? Then you have to ask yourself, which part of the genome are you sequencing, right? So that when you calculate this value, and even if it's different from the expected for the whole genome, maybe for that part of the genome, this is okay, right? Cool.
Généralement, lorsque vous travaillez pour regarder la qualité du contenu GC est très important. Le contenu GC, comment il se calcule? On regarde le nombre de GC qu'il y a dans notre séquence sur la longueur totale. Donc, dans cet exemple, par exemple, le nombre de gènes, c'est 9 sur 2, la longueur des conséquences, ça nous fait 47. Supposons que ce qu'on va espérer, c'est que le contenu GC soit proche. Chaque organisme a un contenu GC. Donc, ce qu'on va espérer, c'est qu'il soit proche de ça. Mais en même temps, il faut faire très attention lorsque vous calculez avec votre contenu GC. Si le contenu GC sur toute la longueur de la séquence, ça doit être très proche. Et si vous séquencez une zone qui est très riche en GC, il peut y avoir un écart par rapport à ce que vous attendez. Donc, il faut faire très attention pour savoir exactement ce que vous faites.
D'abord, pourquoi on s'apprécie sur le GC? Au début, il n'y a pas un intervalle dans lequel on doit travailler. Par exemple, si nous prenons le cas de l'étudiant de l'Europe, il y a un GC équivalent à 47,5%. Et que maintenant, peut-être on trouve 45 ou alors 40. Est-ce qu'on peut conférer cela? Est-ce que nous peut changer
Normalement, on doit s'attendre à quelque chose qui est proche de la valeur théorique. Le proche, c'est pas comment donner, il ne faut pas dire que ça s'écarte grandement. Supposons, je vais pas le cas le plus extrême. Tu t'attends à 47,5 et puis tu obtiens quelque chose de 60. Tu comprends ce que je veux dire?
Question? I think that's difficult. I think in general, you should expect that to be as close as possible. Like, I would say within 1 or 2%, okay? But then again, let's say if you're analyzing, like, let's say 30 samples, if you look at most of the samples, you just know, right? If something is off. Because let's say if you expect 47.5 and most of them are like 47, 47.1, and then you have one sample that is 45. Clearly there's something wrong with that one sample, right? So you should also look at the range of the other samples that you're looking at. If, you know, they're near to what you expect, but you just have a few samples that are different. So that can also help you. All right, so we've talked about number of contigs, assembly length, and 50 value GC percentage. That's a good question. So you could, for example, run something like Kraken. Okay? So something like Kraken will tell you what proportion of your reads are for your target organism. Right? So if you're running Mpox, you would expect that a large proportion of, you know, what you're running are Mpox. Of course, after removing, for example, human reads. But if you get that a large proportion are from another species, and especially if the proportion of your GC is very close, so not too far, the question is it another species within the same genus, for example? Okay? So that is something you have to think about, like, ideally, yeah, I should have talked about Kraken. So Kraken should also help you. It's also one thing that can help you also know whether, you know, your sequencing process is clean. Maybe you have contamination in the lab, or maybe the person who is doing the sequencing is always coughing into the samples, and then human reads are always sequenced, right?
Donc, ce qu'il explique, c'est que quand il y a des écarts comme ça, c'est peu qu'il y ait d'autres micro-organismes qui sont présents. Il y a des outils, il y a des types de Kraken qui ont été utilisés qui permettent de savoir quelle est la diversité qu'il y a de ce que tu as séquencé. Ça peut te permettre de découvrir l'ensemble du process wet lab, tu n'as pas beaucoup de choses.
So I want to finish in 5 minutes. So we want to, you know, this is summary. It's what we call the 4 C's of quality. Number 1, completeness. So the question that you ask yourself is, do you have the whole genome represented in the sequence data? If you're doing partial genome, then of course you'll ask yourself, do you have the whole gene represented in your sample? And here you look at the assembly line. Is it as close as possible to what you expect? So, number one, completeness. Okay?
Donc, lorsque vous regardez la qualité de ce qu'on appelle le cas C, est-ce que le premier C, c'est quoi? Est-ce que la longueur du génome est proche de la longueur de l'échantillon est proche de la longueur du génome?
Right? The second one is contiguity. So here we are asking how broken is your genome. So here you're looking at the number of contigs, you're looking at the N50, and you're also looking at the number of Ns. Okay? So you want this to be as few as possible, you want this to be as large as possible, and you want this to be as small as possible.
Donc, le deuxième C, c'est ce qu'on appelle la contiguïté. Donc, dans la contiguïté, on regarde trois éléments. Le nombre de contigs. Ce qu'on va rechercher, c'est que le nombre de contigs soit le plus petit possible. Le N50, on va rechercher à ce qu'il soit le plus grand possible. Et le nombre de N, c'est normal, on va rechercher à ce qu'il soit le plus petit possible.
Number three is correctness. So, is the assembly correct on a per base basis, and are the reads correctly assembled? So, you look at the average read quality. So, if you have high read quality, then you have better confidence in your assembly, right? You have the average read depth, right? So, the read depth is not necessarily saying that your assembly is correct, but the higher it is, it gives you more confidence that your assembly is correct. Okay? So when you have an average read quality that is higher and an average read depth that is higher, it gives you more confidence that your assembly is also like correct to a higher.
Le troisième, c'est la correctness, c'est l'exactitude. Donc généralement, lorsqu'on veut regarder ce critère-là, on va regarder la qualité moyenne de notre séquence. On va regarder un deuxième élément, ce qu'on appelle la profondeur de couverture. Et donc si on a une bonne profondeur de couverture, ça veut dire quoi? Ça veut dire qu'à cette position, on est sûr que c'est ce qu'on a eu comme à cette position dans notre assemblage chimique. On va regarder deux éléments essentiels ici, la qualité moyenne et la profondeur.
The last one is contamination. So we've talked about Kraken, right? So the question here, are there too many reads from non-target taxa? Or do you have multiple clones? For example, you might have, let's say, you expect your sample to be grade 1B, right? But, you're getting both. I mean, okay, that's not the best example because Kraken cannot distinguish between 1A and 1B. But for those who work with bacteria a lot, sometimes you have classification at the subspecies. So you might end up with two subspecies. You have reads, yes, it's the same species, but there are two different subspecies within your sample. So is there contamination? And you see there's contamination that is caused by you, because you were coughing into the sample, or maybe, which is that common bacteria that contaminates samples in the lab? There's one, the malaria folks, I remember, were always worried about. I think it's usually fungus. There's a fungus. There's always a fungus that contaminates samples in the lab. I don't remember what it is.
Souvent, il y a des champignons sur la paillasse. Des champignons.
Yeah, especially like if you don't clean your pipettes or you don't clean the hood. So essentially, Kraken will tell you if you have that contamination. But you see, there's contamination that is caused by just two pathogens being in the same patient, which is different. It is contamination, but it's a different kind of contamination. Okay?
Le dernier, c'est ce qu'on appelle la contamination. Il faut comment prendre le temps de regarder si vos séquences portent de la contamination. Et ce qui m'explique ici, c'est qu'il y a l'outil qu'on appelle Kraken qui permet de regarder s'il y a de la contamination.
Mais souvent aussi, il y a des bactéries comme Staphylococcus.
You're saying
We have sometimes Staphylococcus which can
yes,
contaminate
exactly. Yes. So obviously, you see, if you're sequencing stuff, aureus, for example, in a sample, you will never know, right? But if you're, let's say you're sequencing C. diff, for example, and you're seeing lots of stuff in other samples, then you need to be worried, like, okay, I think there's contamination in the lab, right? Okay, good. Now, my last slide, yes.
So another common contamination that we've always seen is carryover between micrograms. If you're doing a luminal, you're using the same barcode. Maybe you sequenced clade 1, then you sequence clade 2. So if you're having barcode 1, you have another barcode 1 in the next one. If you did not clean your instrument well, you might see it in the next round.
So it's explaining that contamination could also happen at the machine level, where you have carryover from one sample to the next. Okay?
Do you have any strategies to suggest to prevent that?
So we do, it's recommended that you do a post-run wash. Yes, you do a post-run wash, then multiple maintenance runs just before you do another MySQL run. For us, we use a MySQL. Or you can do, there's a cleanup that you use some percent of Jig to kind of clean up the whole system.
Can someone maybe translate?
Donc, ce qui nous explique ici, c'est que vous pouvez avoir de la contamination. Vous faites un peu de rhum, vous ne faites pas de dégâts, vous pouvez faire deuxième album. Si ce sont des rhums totalement différents, vous pouvez avoir de la contamination. Par exemple, les rhums précédents, le matériel génétique du rhum précédent peut totalement se contaminer. La suggestion ici, c'est qu'il faut faire toute la situation.
Another strategy is that
Vos machines,
It's the same thing we do in the lab. Like, if you're working with RSV, after, you have to clean your pipettes, right? So that the next person who is working with bacteria, they do their stuff clean, right? Then they also have to clean their pipettes. So it's the same with machines. Like, after you do a run, you do a post-run wash, clean everything.
so there's also another, so Illumina now has multiple sets of barcodes. So if you use set A on this one, in the next one you can use B, just to try and avoid the carryover.
Okay?
Can you maybe just add
Like
that?
using different sets of barcodes for different runs.
généralement, il faut utiliser des barcodes différents pour éviter cette situation.
All right, so this is the second last slide. So we've talked about completeness. If you're not getting, a complete genome, perhaps you want to sequence a bit deeper, right? If you're getting a fragmented genome, maybe you want to use long reads because maybe, you just have sections of your genome that are just difficult to sequence. Maybe they could be repeat regions, they could just be structurally difficult. Third, you're looking for correctness. So again, if you're having lower quality, you could try sequence deeper so that you have more confidence in the base calls that you're doing, or even just use higher accuracy sequencing. So for example, if you're using ONT, definitely avoid fast mode and always go for the super accuracy. Or worst case scenario, high accuracy, right? Lastly, for contamination, if it is bacteria, you might want to try and isolate. If somebody just coughed into the sample, you might want to try to resequence, but avoid contaminating the sample in that way. The last slide here, I will remind you, bad data is bad data. So here, they are having the anchor, this one. So this person is saying, if we don't get rid of the anchor, the ship will sink. But this person does not want to get rid of the anchor. It's only got to it. But they will all die. So it's the same thing we often think, like, oh, but I've sequenced this sample, it's expensive, I cannot throw out the reads, this is precious. But you see, bad data is bad data. It's going to lead you to wrong conclusions. So if it is bad, it is bad. There is nothing you can do. Okay?
Ça sert à rien de vouloir garder des états de potentiel. Vous allez tirer de mauvaises conclusions.
Explaining what we cannot track in person.
Sorry?
Tracking that's and explain again with example.
Let me see if I have an example file. I think I should have. Okay. This is a sample output from Kraken. So Kraken will take in all your reads, and then it will tell you what proportion of your reads belong to different species, right? And then, of course, you can see that the classification starts all the way from the kingdom to the species and given to the species level, depending on the classification system of the pathogen. So here, it's telling us for this sample that we have 97.97% of our reads are SARS-CoV-2, okay? Which is good. And down here, we can see that 0.05% are human. So this is good. This is what we expect. But not many samples are like that. So I think I could quickly look through this thing here. Let's see, for example, this sample. Now, if you look at this sample, remember this, we are sequencing SARS-CoV-2. Let me just increase the font. What do we see? Human, what percent? 89. SARS-CoV-2, only 5%. And then we have 0.00 something percent European catfish virus, right? Okay, maybe I don't care about that one. That is too small, right? But of course, I have to ask myself where that came from. But SARS-CoV-2, this is solo, right? Do you think this is you can make an assembly from this one? Do you think it's possible to make an assembly from 5,400 leads?
Vous faites un séquençage et vous avez fait un séquençage pour le SARS-CoV. Et puis vous vous rendez compte que vous avez 5,2% de vos reads qui sont SARS-CoV. Et puis vous avez pour l'humain, vous avez 89,28%. La question qui est posée là, c'est est-ce que vous avez suffisamment de reads pour faire un assemblage? Vous voyez que vous avez en termes de proportion de reads relatifs au SARS-CoV, vous en avez très peu. Voilà, donc ça va impacter votre article dans votre assemblage.
So we always expect that we are going to have some human sequence, right? That happens. But we don't expect it to be this high, okay? Because if I went back here, this particular sample, let me see if we do look at the assembly length that we got. How long is the SARS-CoV-2 genome? 30,000, right? We only got 1,400. You see? So this tells you why. Because we had very few reads that were SARS-CoV-2. There was no way we were going to get a good quality genome from those very few reads. Okay? All right. So now we can go for tea. So, he was asking about. Yes, so we can see how to install Kraken. I think we've also talked about Quast in your environment. Then we can run one or two samples and see the outputs. Is that okay? All right. So, thank you very much. I think that is a good introduction.